today's video lecture. In today's video lecture, I'll be solving previous year's NEET questions and discussing the answers. And this is for the chapter, as you can see on your screen, Biotechnology, Principles and Processes. And we are in part 2 of the series. Alright, so let's begin. Okay, so question number 1. DNA fragments are, and this appeared in NEET 2017, A, negatively charged, B, neutral, C, either positively or negatively charged depending on the size, and D, positively charged. So DNA fragments are A, negatively charged. Okay, so the answer is A. Let's go to the discussion. Now, DNA fragments, as you should know, are negatively charged, and this is due to the presence of phosphate group in the nucleotide. So we know that DNA is made up of nucleotides, right? So it contains deoxyribose sugar plus a base plus phosphate group. And this phosphate group contributes to the negative charge for DNA. All right. So DNA fragments are negatively charged. Pretty simple, direct, right? Let's move on. Question number 2. A gene whose expression helps to identify transformed cell is known as, this appeared in NEET 2017 again. A, vector, B, plasmid, C, structural gene, D, selectable marker. A gene whose expression helps to identify transformed cell is known as D, selectable marker. We already discussed this in the previous lecture also. Please check out part 1. The answer is D. Okay, so here you see a structure of a plasmid, which is a vector for carrying a gene of interest, a foreign gene. And over here you see tetracycline resistant gene and you see ampicillin resistant gene. Now these are selectable markers. Okay, so for example, if a foreign gene is inserted here, now this resistant gene will become inactive, right? That means it will grow in ampicillin antibiotic plate. Okay, similarly it works for tetracyclines. It's the same principle. Okay, so selectable markers are present in vectors to help identify transformants from non-transformants. Okay, so again pretty simple, direct, right? If we go back to the question, yeah, it helps to identify transform cell is known as selectable marker. Okay, let's move on. Question number 3. What is the criterion for DNA fragments movement on agarose gel during gel electrophoresis? This appeared in NEET 2017. A is the smaller the fragment size, the farther it moves. B, positively charged fragments move to farther end. C, negatively charged fragments do not move. D, the larger the fragment size, the farther it moves. Now see, when you're looking at gel electrophoresis, the movement of the DNA fragments is based on the size. It's not based on what charge. So the smaller the fragment size, the farther it moves. The answer is A. Yeah. So here you have a diagram of what gel electrophoresis looks like. So here this gray portion is the gel, and you have the wells over here, and you see the largest DNA fragments are on the top, and then as you move down, the smaller fragments are at the bottom. Okay, the smaller the fragments move down the gel, whereas the large fragments remain on top. Okay, so that's basically the principle. Obviously we know that agarose gel is sieved, so that means the larger ones stay on top, the smaller ones move down the gel. Okay, I think we've answered the question though. Yeah, what is the criterion for DNA fragment movement? That's pretty the criteria. Okay, question number 4. A foreign DNA and plasmid cut by the same restriction endonuclease can be joined to form a recombinant plasmid using, this appeared in NEET 2016. They can be joined to form a recombinant plasmid using D, ligase. Okay, so ligase is the glue. Okay, it joins the foreign DNA with the vector. The answer is D. So ligase enzyme is used to join the foreign DNA and the plasmid cut by the same restriction enzyme to form rDNA. Okay, so yeah, that's the job of ligase. And I think I've discussed ligase also in the previous lecture. It's just the same, it's a repeat of the previous lecture, previous part. That's part 1. Okay, question number 5. Which of the following restriction enzyme produces blunt ends? This appeared in NEET 2016. Now for here, you need to know the restriction sites and how they cut. So SAL1, that's option A, C and D, Hind 3 and XHO1, they form sticky ends, whereas EcoR5 forms blunt ends. So the answer is B. Um, if you, it would be good, I mean, if they ask a question like that where you have to remember which is sticky end and which is blunt end, it would be good for you to remember that's EcoR5, which forms a blunt end, and the rest forms sticky ends. Now, I'm not sure. I think in NCRT they've not really specified all the restriction sites. Maybe whatever is mentioned in NCRT, please study that. Yeah, the restriction site for EcoR5 is below. Okay, so GATATC, that's from 5-3-, and the opposite antiparallel is GATATC. So they are palindromic sequence. Okay, and it forms a cut in the middle over here. Okay, so EcoR5, as you can see, clearly forms a blunt end. It would be good for you to remember if they were asked the question like that in the exam. Okay, but if you go back to the question, yeah, A, C and D options, they form sticky ends, so that doesn't fit the question. All right, let's move on. Question number 6. Which of the following is not a feature of the plasmids? This appeared in NEET 2016. A, transferable, B, single-stranded, C, independent replication, D, circular structure. Which is not a feature of the plasmid? B, single-stranded. So plasmids are double-stranded. The answer is B. A, C and D fit the description. So plasmids are circular, double-stranded DNA molecules. If they are separate from the chromosome, but if you see here, they're actually double-stranded like this. Okay, and then you have your chromosome over here in a bacteria, but this will be separate, it will be independent of the chromosome. And yeah, they're circular. Yeah, they're self-replicating, and they're transferable also. Okay, so these form good candidates for forming rDNA. That's why plasmids are largely used for forming recombinant DNA. Okay, but which is not a feature? Yes, single-stranded. It's double-stranded, we know that. Okay, clear? Yes, we can move on. Okay, so question number 7. Which of the following is a restriction endonuclease? And this appeared in NEET 1 2016. A, DNase 1, B, RNase, C, Hind 2, and D, protease. Okay, so which of the following is a restriction endonuclease? The answer here is C. It's Hind 2. Okay, the answer is C. So Hind 2 is a restriction endonuclease. DNase, RNase, and protease don't fit into that criteria. All right, so let's move on. Question number 8. The introduction of tDNA into plants involves, this appeared in 2015, A, exposing the plants to cold for a brief period, B, allowing the plant roots to stand in water, C, infection of the plant by agrobacterium tumefaciens, D, altering the pH of the soil, then heat shocking the plants. Okay, so the introduction of DNA into plants involves C, infection of the plant by agrobacterium tumefaciens. Okay, so the answer is C. Let's go to the discussion. So we know that agrobacterium tumefaciens is a pathogen, okay, that affects the dicot plants. Now they transfer the tDNA which causes tumors known as T galls. So these bacteria have a tDNA which when transferred to the plants forms crown galls. All right. Now the plasmid, Ti plasmid, induces tumor formation. So this plasmid is called Ti plasmid. Okay. Now the plasmid has been modified, which is not pathogenic. Just imagine if there was no tDNA. Okay, we can use the same concept here. Okay, so imagine if there was no tDNA, the tDNA part was removed, and in place of that, you put your gene of interest over here and then transfer it to the plant. Okay, then you can get your gene of interest into that genome of the plant. Okay, so it's a natural genetic engineer of plants. Okay, so agrobacterium tumefaciens has been used widely in the case of plants because of this reason. Also, the plasmid has been modified. Now, when you remove the tDNA plant, it is not pathogenic. Okay, it is used as a natural genetic engineer of plants. Okay. So, if you go back to the question, yeah, the introduction of tDNA into plants involves this infection of the plant by agrobacterium. Okay. Question number 9. Which vector can clone only a small fragment of DNA? This appeared in 2014. Small fragment of DNA, that's what the question says. A is bacterial artificial chromosome, B yeast artificial chromosome, C plasmid, and D cosmid. So it is C plasmid. Okay, so plasmids can clone small fragments of DNA, whereas A, B, and D, they can clone large fragments of DNA. So the answer is C. So depending on your gene of interest that you want to transfer, you have to look at the size, you have to find the appropriate vector for it. If plasmids don't fit into that, you have to look for another vector where that particular gene of interest can be incorporated into the vector. Okay, so in case of plasmids, small fragments of DNA can be carried. Okay, question number 10. Commonly used vectors for human genome sequencing are, appear in 2014, A tDNA, B BAC and YSC, C expression vectors, and D TA cloning vectors. So commonly used vectors for human genome sequencing are B bacterial artificial chromosome, yeast artificial chromosome. Okay, so the answer is B. They are able to carry large fragments of DNA, so they are useful in the human genome sequencing. Okay, that's what it is. Okay, let's move on then. Question number 11. The colonies of recombinant bacteria appear white in contrast to blue colonies of non-recombinant bacteria because of, this appeared in NEET 2013. A. Insertional inactivation of alpha galactosidase in recombinant bacteria. B. Inactivation of glycosidase enzyme in recombinant bacteria. C. Non-recombinant bacteria containing beta galactosidase. And D. Insertional inactivation of alpha galactosidase in recombinant bacteria. First, let's understand it is not alpha. Okay, so clearly this is wrong, and this is wrong, and this is also wrong. So it is not, recombinant bacteria contain beta galactosidase. Okay, so the answer is C. Let's go to the discussion. Now, the vector in the bacteria contains beta galactosidase gene. Okay, so if you have a plasmid here, there is a gene called beta gal. Okay, it codes for that particular enzyme. Okay, and the colonies of the bacteria appear blue in color when grown in the substrate medium. So now normally if there is no, let's say no insertional inactivation, nothing has been done. Okay, now you plate this on a cultural media, the colonies of the bacteria will appear blue in color. Okay, that's what the nature of this enzyme is. Okay, it forms a blue color. Okay, so you know that there is no insertional inactivation being done. Now let's say if you have this beta gal gene, and an insertional inactivation has been done, a foreign gene of interest has been inserted here, and then you do the same thing, you again plate the colonies over here. Now it will not appear blue, it will appear white in color. Okay, it will appear white in color, so I'm using yellow. to differentiate. It'll appear white in color, so you know that these are the recombinant bacteria. Okay, recombinant plasmids that you have inside the bacteria, all right? So if a foreign gene was inserted, the enzyme gene would be inactivated, and when the colonies are grown in the medium, they would appear white. Okay, hence the recombinants can be differentiated from non-recombinants. Okay, it's a very easy way. You don't have to have use resistant genes, antibiotic resistant genes and all that. For that, you just have one marker, selectable marker, which is the beta gal marker, and you just insert the foreign gene that you want here, and you can directly plate it in the media, and you can see whether you have got the whether it has been transformed with the foreign gene or not. Okay, after you have transformed into the host cell. Okay, so that's how it works. We go back to the question. Yeah, the other options don't make sense. It's C. Non-recombinant bacteria contain beta galactosidase gene. All right, let's move on. Question number 12. DNA fragments generated by the restriction endonuclease in a chemical reaction can be separated by, this appeared in NEET 2013. They can be separated by A. electrophoresis, B. restriction mapping, C. centrifugation, and D. PCR. So, DNA fragments generated by the restriction endonuclease can be separated by electrophoresis. Okay, so the answer is C. We already discussed electrophoresis. It's pretty much the same thing, so I'm not going through this. I've already discussed this in part 1 also, what electrophoresis does. So after you have treated with your restriction endonuclease, restriction enzymes, your plasmid has been divided into different fragments. Now you have to select which fragment you want, or you want to view how it has been cut. So you run a gel, and you can see the large fragments, you can see the small fragments at the bottom. Okay, and they are separated based on size. So this is how it is. All right, let's move on then. Question number 13. The given figure is a diagrammatic representation of the E. coli vector PBR 322. Which one of the given options correctly identifies its certain components? This appeared in 2012. A. ORI, original restriction enzyme, no. B. ROP, reduce osmotic pressure, no. HIND3 and ECO-R1 selectable markers, no. Ampicillin R and tetracycline are antibiotic resistant genes. Yes, answer is D. Okay, so that's the correct answer. The answer is D. So these are the correct answers. ORI is origin of replication, ROP is repressor of primer, HIND3 and ECO-R1 restriction enzyme sites, ampicillin resistant, tetracycline resistant tetracycline resistant genes are antibiotic resistant genes. Okay, so that is the correct option. Okay, clear? We've answered the question here. Let's move on then. Question number 14. A single strand of nucleic acid tagged with a radioactive molecule is called, this appeared in 2012, A. vector, B. selectable marker, C. plasmid, and D. probe. So the answer here is probe. Okay, answer is D. Now probes are what? Single-stranded DNA or RNA molecules that attach to the desired DNA or RNA fragments and are identified when exposed to X-ray film. Okay, so now what happens is, once you have run the gel electrophoresis, let's say you want to find a particular DNA fragment. Okay, you run the gel electrophoresis. Now there is a method called southern blotting. Okay, there's a method called southern blotting, and what it does is it forms like a photocopy of this gel on a nitrocellulose paper. Okay, let's say this is a gel, this is the nitrocellulose paper. Now, I'm not going to the details of how southern blotting is done, but just understand that it's like a photocopy of the gel, okay, in on a nitrocellulose membrane, okay. And this particular membrane is then placed in a solution in which there are lots of probes added to it, okay, lots of probes added to it. Now there will be some probe that will match or form a hybridization with the DNA fragment here, okay. There will be lots of probes in it, and there will be some probe will go and match over here, okay, will go and match over here. All right, now this particular probes, they are either fluorescent label, or they can be, like it says over here, they are radioactive as per the question. It's not written here, sorry. Okay, they can be let's say you have a single stranded DNA over here, and they are either fluorescent labeled or radioactive, sorry, radioactively labeled. Okay, let's say they're radioactively labeled, okay. Now it will attach to a particular DNA sequence. Yeah, it will attach to a particular DNA sequence in this on this membrane. Now you want to view it. Obviously you can't just view it like that with a naked eye, right? So you put it under an X-ray film. You put it under an X-ray, and you can then view that particular DNA fragment there. So you know that that particular DNA fragment is present there, okay. It's used for identifying pathogenic diseases or cancers, you know, things like that. That's usually the application. So probes are very useful in that, okay. They can help you identify exactly where that particular DNA fragment of interest that you need is there, okay. So they are fluorescently labeled or they're radioactively labeled, and then they can be put under an X-ray film, and you can view that particular fragment over there. Sorry, I don't know what's wrong with this pen. You can then view it under an X-ray film, that particular DNA fragment, okay. So this is how probes work. All right, so but to answer your question, it's pretty direct. Probes are single stranded and they attach to the desired DNA fragment or RNA fragment. For that, for RNA fragment you do northern blotting. For DNA fragment, DNAs you do southern blotting. For proteins you do western blotting. For that, antibodies are used to identify particular proteins. It's called western blotting, okay. Again, the antibodies are fluorescently those probes, they use as probes, and they are fluorescently attached or radioactively attached, and then you can view that particular protein of interest that you want, okay. So basically this is what it is. We look at the question. Yeah, the answer is D, probe, okay. All right, let's move on. Question number 15. For transformation, microparticles coated with DNA to be bombarded with gene gun are made up of, this appeared in 2012, A, silver or platinum, B, platinum or zinc, C, silicon or platinum, or D, gold or tungsten. Okay, so the answer is D. Okay, the answer is D. So for transformation of cell into the host, I'm sorry, transformation of DNA into that host, there is a method called gene gun, okay, gene gun, okay. And in that there are microparticles which are coated with that DNA, and these microparticles are made up of gold or tungsten, okay. They're pushed into that host cell, okay. So it's called gene gun. It's like a gun, it's pushed into it, okay. And those microparticles are first coated with the DNA that you want to go inside the host cell, and then they are pushed into the host cell. So they are made up of gold or tungsten. Yep, the answer is D. The others don't fit the criteria. All right. Question number 16. Biolistics or gene gun is suitable for, this appeared in Main's 2012. A. Disarming pathogen vectors. B. Transformation of plant cells. C. Construction rDNA by joining with vectors. D. DNA fingerprinting. So, bialistic or gene gun are suitable for transformation of plant cells. The answer is B. Okay, they are suitable for transformation of plant cells. So remember that this information there in NCERT also. So no excuses for not knowing that. Okay, clear. Let's move on then. Question number 17. In genetic engineering, the antibiotics are used, this appeared in Main's 2012, as selectable markers, B to select healthy vectors, C as sequences from where replication starts, D to keep the cultures free of infection. So antibiotics are used as selectable markers. Okay, the answer is A. So antibiotics are used in the plating method to allow differentiation between transformed cells and non-transformed cells. Alright, let's say now you know that you have a plasmid and you have the antibiotic resistant gene. Okay, that means it's resistant to that particular antibiotic. Let's say you have a tetracycline resistant gene over here. Okay, now tetracycline resistant gene will grow on a plate containing tetracycline, which is resistant to it, right? It does not affect, it has not been affected by the antibiotic that is present over here. It's resistant to it. Okay, so it will grow. All right, so let's say if we add our gene of interest over here, okay, and this inactivates tetracycline resistant gene, then what will happen? You do the same method and you plate the bacteria over here, it will not grow, right? Because now this tetracycline resistant gene has been inactivated. All right, so that's how antibiotics are used to differentiate transformed cells from non-transformed cells. All right, okay, let's move on then. Question number 18. Which one of the following represents a palindromic sequence in DNA? It's appeared in Main's 2012. So you have your options A, B, C and D. So you have GAATTC, GAATTC. So clearly it's option A. The rest don't match. It's not C. And it's not D also. So the answer is A. Alright, so option A represents a palindromic sequence. Okay, so it works this way and works backwards also. These are called palindromic sequences. Alright, let's move on. Question number 19. Given below is a sample of a portion of DNA strand giving the base sequence on the opposite strands. What is so special shown in it? This appeared in 2011. So you have GATTC, GATTC. Clearly looks like a palindromic sequence. So you have A, replication method, B, deletion mutation, C, start codon at the 5' D, palindromic sequence of base pairs. So it's palindromic sequence of base pairs. The answer is D. Okay, we just discussed palindromic sequences, so I won't repeat this again. Question number 20. There is a restriction endonuclease called EcoR1. What does CO represent? Part in it stand for? What does it represent? It's appeared in 2011. A colon, B coelom, C coenzyme, D coli. So clearly it is D coli. Okay, it is E. coli. So CO represents coli and EcoR1. This E. coli genus and species. RY13 is the strain and 1 is the order of discovery. That's EcoR1. Alright, let's move on then. Question number 21. Agarose extracted from seaweeds is used in, this appeared in 2011. A. Spectrophotometry, B. Tissue culture, C. PCR, or D. Gel electrophoresis. So agarose extracted from seaweeds is used in D. Gel electrophoresis. Okay, that's where agarose comes from. The answer is D. So it is used in forming gel for gel electrophoresis. Clear? This was pretty direct. Okay, and we've come to the end of this video lecture, and I hope you've enjoyed today's video lecture. Hope you've learned something new today. And don't forget to like, share, and subscribe to MJ NEET Biology. And I'm also available on Facebook, Twitter, and Instagram, so you can follow me there for more updates. All right, and I'll see you in the next video lecture. So, bye.